All right, it is 3 o'clock, so let's get this session underway. First of all, I'd like to thank you guys for coming out to this session. I know it's the weekend and you probably have lots of plans or, homework to do, stuff like that. So thank you for setting aside time to come to this session. And I promise you, you won't regret it. So why don't I share my screen and we can get underway. Right. So, this is the only alternative virtual SI session that I'm going to be able to hold before you guys have your second midterm, because that's going to be not the coming Tuesday, but next Tuesday or Monday, I believe. So I figured that I would make an agenda based on all the things that have been covered so far. So this is pretty comprehensive, which means we have a lot of stuff to go through. But hopefully, once we come out at the end, you guys will feel a lot more confident about the material. But before we actually get into that, I'd like to set us up with some alternative VSI expectations and rules, just because this is a little bit of a different format. And then we can move on to the study guide terms. And in the last 5 minutes or so, I'll answer any questions that you guys have at the end. So yeah, alternative VSI, it's not exactly the same as regular SI sessions because I do need you guys to stay muted the entire time, and I also need you guys to keep your cameras off the entire time. And this is simply to protect privacy of individuals so that we can upload the recording of this video to YouTube later for access, wide access. But that doesn't mean that you can't ask me questions or answer questions when I ask you guys stuff. You'll just have to leave that in the chat. And I will also monitor the chat so that none of that goes over my head. Additionally, you guys will get access to session materials as soon as they're available if you attend sessions, just like you are doing right now. And other students will only get access to these materials, 1 to 2 weeks before the midterm. Now, because this session is actually less than 2 weeks before the midterm, everyone is going to get access to the materials as soon as they are available. But I will probably share the slides for today with you guys immediately after the session, just because I think they're very handy. So in a sense, you'll get that bit of a head start this time. But yeah, the materials include the slides that I use. If I ever use documents, that will be shared. And of course, the recording of the entire session. And the great thing about the recordings are they come with timestamps, and they cut out any of the unnecessary bits. So you can fast forward to whatever piece of the recording you think would benefit you the most. And another disclaimer is that if you guys are attending the sessions, obviously you're going to get early access. And I would really appreciate it if you guys don't share the materials with other people, even if they're your friends and they're like, hey, please, can I get this material? Because, it negates the reward for those of you who've actually set aside time on the weekends to make it to these sessions. So just something to keep in mind. But So yeah, as long as you guys understand, I guess we can, get on with the materials at hand. And so, oop, um, essentially what I did is I broke the studying down into 4 different slides or themes because I feel like, there are certain material concepts in the class that are more closely related to each other. So that's kind of how this is separated off. And I made sort of like a guide to how you should navigate the terms as well. So the first thing we're going to do is we're going to start off with photosynthesis and photorespiration. So um, obviously, at the beginning of the unit, you guys covered photosynthesis, and you know that photosynthesis is made up of 2 parts, the light reaction and the Calvin cycle. So we're going to dissect those separately first and just hit the main points, the things that you really probably should know. Like there's some specifics that are, they'll help you in your knowledge, but are probably not as important for studying for the exam and probably will more distract you than help you when you take the exam. So I just made a list of all the important points that we need to tackle. So obviously for the light reaction, and that's where we're going to start here, you have some more important terms like the granum, which is a stack of thylakoids. And if you remember, the thylakoids are the sacs where the light reactions happen, as well as the thylakoid membrane, where the light reactions happen along. So like the stroma is on the outside and you have the thylakoid, and along the thylakoid membrane, you have the light hitting the electron transport chain and all of that happening. And the big breakdown of the light reactions, as we know, is photosystems 2 and 1. And if you remember, the confusing thing about photosystems 2 and 1 is that photosystems 2 actually comes before photosystems 1 because photosystems 1 was the first part of the light reaction that was actually discovered by scientists. But essentially what happens is in photosystems 2, light hits the thylakoid membrane and excites the electron and gets it to a certain like state of energy. And this pushes the electron through the electron transport chain and allows for hydrogen ions to come through, basically to come through the thylakoid membrane. It opens up passageways so that hydrogen ions can come into the thylakoid membrane and separate from oxygen, which is a waste byproduct. And then in photosystem 1, the excited electron continues from more light hitting it, and this time the energy actually helps NADP plus to become NADPH with also the hydrogen, some of the hydrogen ions, and the energy from photosystem 1 making that combination. So those are the important things to remember about each photosystem. And how the photosystems work are obviously you have light hitting the thylakoid membrane, but more specifically, you have the antenna complex, which receives the light. It's basically like a receptor, and it has like chlorophyll A, and it takes the most energy from blue and red light because those are the most energetic in that sense, and it reflects green light and a little bit of yellow light mostly, and that's why green plants get their green color from. And then the energy is trapped in the reaction center, and the reaction center is actually what's used to power the electron transport chain that sends the electron across and activates the hydrogen ion channel into the thylakoid membrane and activates the NADP plus conversion to NADPH. So that's the big rundown of the light reaction. Obviously, then the second part of photosynthesis is we have the Calvin cycle. And so the Calvin cycle happens in the open space of the chloroplast, which is the stroma. And you start out with a compound 1,5-ribulose biphosphate, basically a 5-carbon molecule. And what's happening is you have ribulose biphosphate, which is just a substance, and the cell takes in CO2 from the atmosphere. And basically, you have this enzyme in the Calvin cycle called Rubisco. And the role of Rubisco is to fix carbon, or essentially what that means is to take the carbon from carbon dioxide and add it onto ribulose biphosphate to make a 6-carbon sugar, at which point the 6-carbon sugar is broken down so that you have G3P, and the sugars can be used for other processes in the cell, or it can be used to continue the cycle, essentially power the plant. The sugars, that's the purpose of the sugars. And then you have, your ATP and your NADPH that you got from the light reaction. And sorry, I skipped over this. The ATP is produced from the ATP synthase when you funnel hydrogen ions through that tunnel at the end of the light reaction. And so you use ATP and NADPH to break down the 6-carbon molecule into G3P, and what you have left over is ADP and NADP plus, and those two things go back to the light reaction to be converted into ATP and NADPH again. So that's why they're kind of recycling off of each other. And then obviously photosynthesis is really great, but then you have this danger to photosynthesis, which is actually photorespiration, which specifically affects the Calvin cycle. And how the Calvin cycle works is when carbon is added, when Rubisco helps carbon add onto the ribulose biphosphate, that's when you make the sugar, and that's when the process is normally working and helping the plant get energy. But if you have oxygen introduced and the Rubisco fixes the oxygen to the ribulose biphosphate instead, you get a waste product which is essentially useless, but then the cell also has to spend energy breaking down the compound that's created. And so, obviously, this is a major problem when when carbon is not readily available in the air, like it's not super abundant. So that's when oxygen is able to come in and affect the Calvin cycle. So obviously, if you're a plant, you want to bring in carbon to fix that problem. But then the other issue you face is, and especially in hot areas and dry areas where there isn't a lot of water and you need to retain water because you could easily lose it, you have the problem of transpiration, where if you open the stomata of the plant to bring in carbon dioxide from the atmosphere, you're also letting water out in vapor, and you're losing that, basically dehydrating the plant. And that's a big drawback. So obviously, as a plant, you need to make sure that you get in as much carbon as you can to continue photorespiration without losing a lot of water to the point that it would be detrimental or really harmful. So, and the typical plant, the C3 plant, doesn't need to do anything because it's in a warm area, which also is wet, has lots of moisture or is humid, and basically there's enough water in the air and not enough heat where the photorespiration really becomes a problem for the plant. But in hot areas and dry areas, you need a solution. And the solutions are the C4 plant and the CAM plant. And so the C4 plant, how does it work? Essentially, you have the stomata, which usually you would open up and you would let carbon dioxide come in and you would lose water as well. But a C4 plant, instead of doing that, it has a different way of getting the carbon that it needs. So it keeps its stomata closed, so it never loses water, or it doesn't lose a lot of water, to the heat. And the way it absorbs carbon from carbon dioxide is actually taking carbon dioxide from the air and absorbing it in the mesophyll, essentially turning the carbon from the carbon dioxide into a 4-carbon compound. And this 4-carbon compound, why it's created is so that the carbon dioxide can be moved into the plant and bypass the stomata without the stomata opening up. So that's why the C4 compound is created. But obviously, the C4 compound in and of itself is not very useful for the plant because it needs carbon from carbon dioxide for Rubisco to actually latch onto it and fixate it to create sugars. So the C4 compound is converted back into CO2 in the bundle sheath cells in a process known as decarboxylation. And so that's what is able to get the carbon from the air into the C4 plant Calvin cycle without opening the stomata up and without losing a lot of water. The only issue with the C4 plant is because you're doing so much to convert carbon into a C4 compound and then turn it back into CO2 to be used in the Calvin cycle, that requires a lot of energy. So it is energy consuming, energy taxing, energy heavy. Whereas you have this other alternative called the CAM plant, and you probably won't need to know this, but it stands for cruciation acid metabolism plant. It's very wordy, a big mouthful. And essentially, the CAM plant's solution to this is that instead of opening up the stomata during the day to get carbon dioxide and while losing water and then closing up in the night, the CAM plant reverses the regular C3 plant process by opening itself up during the night when it's not so hot and it won't lose as much water. And basically it can absorb all the carbon dioxide from the air and it doesn't lose too much water. And then it stores that carbon dioxide as a C4 compound in malic acid, which is internal in the cell. So similar to the C4 plant, it needs to convert the carbon dioxide to a C4 compound. And so it stores C4 in the malic acid. And then when it's the day, the stomata closes again so that you're not losing any water and you don't need to absorb any more carbon dioxide because you got that during the nighttime. And again, similar to the C4 plant, you have a decarboxylation of the C4 compound that's stored in the malic acid. And essentially, what that happens is the C4 compound in the malic acid is converted back to CO2 and used in the Calvin cycle. And so Mai asks, so which are the cacti, C4 or CAM plants? So cacti are usually going to be CAM plants, and that's just because CAM plants reside in areas that are hot and dry. So obviously the desert is a hot and dry environment, so a CAM plant is more suited for that area. C4 plants are meant for hot areas, but hot are also maybe humid and not so dry. Yeah, so no problem, Mai. So that's basically a rundown of photosynthesis and photorespiration. And I know that's a lot, but we got through a lot. And I hope this guide helps you figure out how to navigate all the material when it comes to photosynthesis and photorespiration. All right, why don't we move on to the next subject, which is plant growth and development. So obviously this is the lecture that Dr. Wilkin had right after she covered photorespiration and photosynthesis. And so I broke this one down into 2 separate parts as well, plant growth and plant development, because there is a very important distinction between them. So why don't we tackle plant growth first? So plant growth is essentially how the plant develops in its later stages. This is after it was fertilized as a seed and after it like initially set itself into the ground. This is now that the plant is like a baby, how does it grow from there, and how do the cells differentiate from each other? Why do they gain certain functions and stuff like that? So, but obviously you have different types of cells with different types of growth patterns. So for example, indeterminate growth means that the cell of the plant does not necessarily have a written function in it, and that it'll actually be able to adapt to the surroundings as it grows. And that's pretty good. Uh, Root apical meristem cells at the bottom of the plant essentially push into the ground, allowing for the root apical meristem to uh, dictate growth underneath the plant without being destroyed by the force to push into the soil. And then you have other parts on the plant, like the axillary bud, which is for the growth of the leaves and the branches of the plant, and essentially is lateral growth uh, from from the stem, but not within the stem. So axillary buds, they mark lateral growth from the stem. And then you have different parts of the stem as well. And I kind of made uh, a line of the different parts of a typical plant stem so that it's easy to remember in what order they go. So epidermis, that's the most outer region of the plant, basically the lining on the outside. It's the protection. Then you have the cortex, which makes up most of the inside cells of the plant, basically just for structure and what makes up the innards of the plant. Then you have the primary phloem, which is obviously for distributing sugars within the plant, specifically bringing sugars down from the leaves to the roots of the plant and down below. You have the xylem, which transfers water and nutrients from the roots of the plant from the bottom up to the leaves that need it. And then you have the pith, which is kind of the main stem, main transport passage of the plant that also serves as structure, which is essentially the middle of the plant. So as long as you remember this order from out to in, that's essentially how a regular plant stem looks. So just keep that in mind. Then you have secondary growth, which is epidermic bark then. So bark is actually not the typical epidermal tissue. It's more of an external barrier. It is epidermal in the sense that it is like related to the tissue, but it's just not what you would associate with the regular epidermal tissue of a regular plant. So, but yes, for trees, it is a form of epidermal tissue. So yeah, then we go into secondary growth, and secondary growth is specific to wood plants and specific to eudicots. So outside of eudicots and wood plants, regular plants do not have secondary growth. And so what is secondary growth? Secondary growth is lateral growth or horizontal growth, but different from the axillary bud, which is growth from the stem outward into branches and leaves. Secondary growth is growth in the stem outward. So basically widening the base, widening the trunk of a tree, stuff like that. And so in secondary growth, essentially what you have is secondary pieces or a copy of each of the terms that we looked at above: epidermis, cortex, phloem, xylem, pith. You basically have another one of like the phloem. You have a secondary phloem and you have a secondary xylem, secondary epidermis, which essentially builds up the extra layers of the cell of the plant trunk and base as it grows outwards. And then you can also remember the vascular cambium, which is kind of like serves as the line that defines the separation between the primary growth section and secondary growth section, and like consecutive growth sections. So the line that divides them, you can refer to that as the vascular cambium. And then obviously, yes, for trees, you have bark on the outside, which is an extra protective layer. And just like Mai pointed out, it is another form of epidermal tissue. So something to keep in mind. Then we go on to plant development, which honestly, maybe I should have covered plant development first because it actually affects the earlier stages of a plant. Basically, what you have is plant development covers gametogenesis and embryogenesis. Gametogenesis is when you have the gametes of a plant, and essentially, this is the process in which the gametes, come together and fertilize the egg or the seed or the offspring of the plant. So that's like very early stages, essentially pre-formation of the plant. Then embryogenesis, you have the plant that is fertilized, and now we're talking about germination, which is the early settling, essentially establishing your roots, establishing your axes, or how the plant will grow from there on. And so there's a couple things that embryogenesis establishes, and I mentioned axes, and there are two axes that you need to remember. So the radial axis is the axis that determines how the plant grows out in the base, so essentially horizontal within the base. And then the apical basal axis is how the plant grows vertically, up and down. And an easy way to remember the difference is apical meristem. We know apical meristem is related to growing from the shoot and growing from the roots. So that's an easy way to remember the apical basal axis. And then all you need to remember is that the radial axis, you can take the radius from the center and it just goes outward. So that's a good way to differentiate between them. Then you have the tissues. So obviously embryogenesis establishes the three tissues of the plant. You have your epidermal tissue, which is protecting you from the outside. You have your ground tissue, which essentially establishes structure and establishes, how the plant stays in the ground. And then you have your vascular tissue, which is very important for transporting nutrients, sugar, water, and the important vascular tissue to remember are xylem and phloem, which we talked about earlier. So yeah, that's the tissue that's established. And then it does also establish your shoot apical meristem and your root apical meristem, so that the plant will grow from there. It just doesn't dictate the growth of those two itself. It just sets it up. So yeah, cool. All right, that's 2 down and 2 more to go. I hope you guys are following along, and don't be shy to let me know if you guys have any questions as we're going. So next subject, we've got plant hormones and responses. So hopefully you guys are fairly refreshed on this because this was earlier on in this week. But yeah, essentially I divided it again, plant hormones as one and plant response as another, because plant to determine how the plant will grow, behave, germinate, develop, stuff like that. It's pretty straightforward. Then you have signal transduction, and essentially that's like different molecular events, whether they be chemical or physical, that help the plant communicate within itself, within its different parts, what needs to be happening with the plant hormones and how they need to be used. And for every signal, you have, first of all, a stimulus or stimuli, which essentially are what the plant reacts to and creates the signal from or for. You have the receptor, which acknowledges the stimuli or stimulus, and basically is what alerts the plant that there needs to be some action. And then obviously the relationship between the stimuli and the receptor results in some sort of tropism, which is a movement towards or a response to a stimulus or stimuli. And so in plants, there are two big stimuli, which are obviously light from the sun. And then we also have gravity, which affects all of us. And so phototropism essentially is that the plant uh, detects the stimuli of light when within its surroundings, wherever it is. And as a response in its receptor, it will move closer towards the light to make sure that it can get as much light as possible. And this could mean anything from, going more vertical or stretching farther out to get more distance between your leaves, or even bending, which we covered as an example in lecture, where you could have auxin going to the dark side of the plant to enable elongation in the cells in the back so that the plant can bend towards the direction that it needs. And often this is in response to light, where you want to get closer to the light and you want to get more light, and they want to grow into uh, branches, and they want to grow into leaves and like spread out like that. And so the uh, the lateral bud is delayed, and especially when you have auxin at the top of the plant, that is specifically enforced. Then you have the next plant hormone, which is cytokinin. And cytokinin is all about cell division, and that's pretty easy to understand when you think of cytokinin, cytokinesis. Cytokinesis is the process through which the plant cells divide. And so that's like the big thing about cytokinin. But it also helps with lateral bud growth, with xylem growth, and with stomata opening. So it's really about development of the plant in specific avenues. Obviously, developing the branches, the leaves, developing the water nutrient transport system, and opening the stomata up so that the stomata can absorb carbon dioxide from the air, and also hopefully the stomata doesn't lose too much water. Then it also delays senescence, similar to auxin, because it's more of a developmental plant hormone. So as long as you have auxin or cytokinin, you're kind of putting off the natural aging of the plant and the deterioration of the cells and the ability to divide. And cytokinin is also really useful for the response to immediate stress that the plant might receive. And by immediate stress, I don't mean like if a caterpillar starts eating the leaf, the plant's suddenly going to whack the caterpillar or something like that, right? That's like not how the plant system works. It takes a lot longer to respond to threats like that. But immediate stress is sort of like a caterpillar is eating the leaves, but then as we discussed, the plant can emit some sort of scent or some sort of signal that attracts other um, organisms that will actually prey on the caterpillar or take advantage of the caterpillar so that the caterpillar doesn't feed on the plant anymore. So that's kind of the immediate stress response that cytokinin can elicit. Then you have gibberellins, which is another growth hormone. It really helps with stem elongation, and more specifically internode elongation, or the internode is the area between two nodes or branching out points. So specifically increasing that area, gibberellins is really helpful. Gibberellins also enhances germination, so early development of the plant, and flowering of the plant or development of fruits in the plant. So it's really good for both early and late development in that sense. Then we move on to abscisic acid. And abscisic acid, unlike gibberellins, unlike auxin, unlike cytokinin, is much more of a barrier hormone. And I say this because it does a lot to slow down plant processes, specifically development, or sometimes it can actually speed up plant deterioration. So an example of this is abscisic acid. It makes sure that the stomata will close, and this can be good if you're losing a lot of water. It also delays root development or root spread, so the plant won't be able to get as many nutrients or reach into the soil as much, but this can be beneficial when the soil actually isn't suited for the plant. Like you might be in a off season like winter or something where there's not much to go off of, and so you don't want to spread out and waste energy. And abscisic acid promotes senescence, so it essentially speeds up the process of aging and the loss of the division power in cells. And it also promotes dormancy in early stage plants. So once you're a fully developed plant, it's not really going to be an issue. But when you're like a seed, the dormancy can actually help the seed not sprout during an off season like winter, where it won't be able to survive. It'll probably perish or die off because of the harsh conditions. And it can actually grow in a better season like spring or summer, where there might be more rains, more sun, more nutrients in the soil, stuff like that. And then our final plant hormone is ethylene. Unlike the other plant hormones, this one is actually in a gaseous state while it is in the plant. And so that does give it different properties. It also makes it a lot easier to transfer around. And specifically what it does, it's kind of like a mixed bag. It's not like abscisic acid where it completely delays processes, and it's not like auxin, cytokinin, or gibberellins where it kind of speeds up all processes. Ethylene kind of takes the best of both worlds, or a little bit from both worlds. So in terms of delaying stuff, it delays stem elongation as well as the auxin transport within the plant. So it is a hindrance to both of those things. And then it also, on the other hand, promotes fruit ripening. And this can be good, but sometimes when you have fruit ripening, it accelerates it very quickly to the point where it actually goes rotten really quick. So too much ethylene can obviously be a problem if you're trying to grow fruits and you don't want them to go bad. And then ethylene is really the hormone that's responsible for plant response to environmental stress. And by environmental stress, this is very different from the immediate stress I talked about earlier, where essentially it's what is the condition that your plant is residing in. So what's the weather like? Are there like fires? Are there hurricanes? Are there heavy rains? Essentially, what is the plant dealing with that's coming from the environment around it? So that's what ethylene is really responsible for. And so all of this, some of the things that we talk about when we're talking about immediate stress, going back to cytokinin, immediate stress, is that you have things like proteinase inhibitors and green leaf volatiles. And essentially what these are things that are secreted on plants such that, so protein inhibitors make the plant basically undigestible for organisms that eat it. Basically the enzymes that the organisms have cannot break down the plant, and therefore the organisms will probably stop going to the plant to eat it because it's not helpful, or you have green leaf volatiles, which similarly do bad things to organisms that threaten these plants, such as the example that Dr. Wilkin had about signaling the wasp and then the wasp stinging the caterpillar so that the babies of the wasp would use the caterpillar as fodder for growth in their early stage. So that's kind of what a green leaf volatile is. It's something that helps the plant to deter danger that affects the organisms. And it's less specific than the proteinase inhibitors, which specifically target the digestibility of the plant itself. All right, so it looks like we are on to our last subject, and thank you guys for holding on. And you don't have water to help you do that, or a lot of water to help you do that, and you're fighting against gravity sometimes to get nutrients and other materials to specific parts of the body. So then we break it down into two different types of plants. We've got vascular plants and non-vascular plants. And so I'm actually going to cover non-vascular plants first because they are probably an earlier development from the green algae. And essentially, non-vascular plants, you don't have any vascular tissue on land to help your nutrients get around or to essentially give you a lot of distant support. And so the way you get nutrients traveling around is through diffusion. And diffusion can only be done if you are near the water because you need some sort of liquid substance to make materials travel through. And so the limitations of using diffusion to transport materials is obviously you need to be close to water. And also you don't have a lot of size potential for your plant. You can't really spread out that much just because you don't have a wide transport system. So those are the limitations of diffusion. And within these plants, you'd have two different stages of reproduction. You have the haploid gametophyte, which is essentially the gamete that is going to be part of the offspring, but only makes up half of it. And so that's part of your life cycle phase where you're creating the gametes that will be necessary for your offspring. And then you also have your offspring, which are the diploid sporophytes. And those will be what actually grow up into full plants and the next generation. And the sporophytes release spores in these non-vascular plants to get their offspring situated elsewhere and to make sure that they can grow up. And the main plant that we actually look at under this category is bryophytes. And bryophytes, they have a haploid dominance, which is unlike any of the other land plants we're going to look at, because all of the other ones have a diploid dominance in them during their life cycles, and that's because there's a gradual evolutionary shift towards that in plants. And so at the bottom, I kind of put another one of these arrow things here, these lines, to think of when you're thinking of alternation of generations. Angiosperms have the largest diploid phase. Gymnosperms have the second largest diploid phase. Then lycophytes. And lycophytes actually have diploid and haploid phases that are independent of each other. And finally, you have the bryophytes, which actually have a haploid dominant phase. So that's a good thing to remember. That order is probably useful. All right. So we covered non-vascular plants. So why don't we look at vascular plants real quick? Obviously, to transport nutrients, you have the xylem and phloem, like we covered. Xylem transports water and nutrients. Phloem transports sugar. Then you have the idea of, okay, now you have a vascular plant. How are you going to transport your offspring? Are you going to use seeds or are you going to be seedless? So seedless plants, that actually covers another group called the lycophytes or the ferns. And essentially, similar to the bryophytes, they spread their offspring by using spores. And then we look at our seeded plants, which are more common, at least to what we know. And the benefits of having seeds is that you can use a gamete transfer, but it's completely waterless. You don't need to rely on water to get your offspring anywhere or to keep your offspring healthy and ready to grow somewhere else. And so the two main plants that derive from the seeded vascular plants are the gymnosperms and the angiosperms. And so gymnosperms, the important difference is they have naked seeds, so there's no encapsulation around these seeds, and they do not sprout flowers or fruit. And then obviously angiosperms, because we're talking about differences, they do have enclosed seeds and they do produce flower or fruit. And so those are important things to remember when you're talking about the distinctions between all these different plants. And this chart, I think, helps really categorize how this works. So Mai asked, what is an example of a non-vascular plant? Essentially, like you have the ground plants that reside near water, similar stuff that's similar to algae. You have like mosses, liverworts, I believe, stuff like those stuff. Plants that don't really have a lot of size or a lot of structure and are very loosely developed. So yeah, mosses, liverworts, and there was one other I can't remember right now, but those three different groups make up bryophytes and non-vascular plants. But yeah, that's pretty much all of the material that you guys have covered in plants in class about plants so far. I hope you guys understood kind of where my thought process was going with how I set up the study guides. I will share the study guides with you after the session is over. But before we go, do we have any questions? And Kayla, I see that from you, so I'll take a picture. But yeah, do we have any questions before I let you guys go to enjoy your weekends? How important is it to memorize the parts of the plants? So I'm assuming you're talking about how we talked about the epidermis, the cortex, xylem, phloem, and the pith. Is that right, Mai? Yeah, no problem. So what I would say is the most important parts to memorize about the parts of the plant, just really know what the xylem does, what the phloem does, and understand that the epidermis is essentially the protective skin of the plant. Outside of that, I wouldn't stress too much about the makeup of the plant because that's really not so important to how the plant functions, right? The xylem and the phloem come up so often, and so that's what you should really think about. Yeah. And of course, it's my pleasure to host a session for you guys. Um, you know, as much as I can do to help you guys study better for the exam, that's what I'm here for. All right. Any other questions that you guys have? Sure, sure, sure. I'm always happy to help. Yes, you guys too. Have a great weekend. Relax, take some time off. Absolutely, no problem, Fook. No problem. Thank you for coming, Fook. Thank you. Do you have any other questions for me, Fook? Absolutely. Of course, that's what I'm here for. Alrighty. Yes, no, absolutely, no problem. All right, if you don't have any other questions, I think I'm gonna call the session here. Thank you so much for coming again, Fook. I will see you on Tuesday.